So thanks everyone to be on time. It's my real pleasure to introduce the first speaker of today, who is Professor Hamid Reza Marateb. So Hamid is currently professor at the University of Isfahan, but also at UPC, and as you've seen, also affiliated with the Smart University of Medical Science in Tehran. And yes, and you may know Hamid. For those in the field, for the development of the EMGLab software, along with Professor McGill. So Hamid has done a lot in EMG signal processing, in EMG decomposition, and is now very active in data mining and statistical analysis. But we want to get his expertise on EMG decomposition today. So this is what the talk is about. I think the sound is good, but if not, I can increase or decrease the volume. So, Hamid, the floor is yours. Thanks.
Okay.
Setting to the school.
Okay, thank you very much. Hello, everybody. Thank you, Sylvia, for inviting me, and a very nice introduction. Yes, I have been working on EMG decomposition since 2003, I guess. And we developed, Kevin and I developed EMGLab, and it has been used in many laboratories as an open source program. Now, I'm going to talk about template matching based decomposition, and I will focus a kind of top-down strategy, talking about the basic concepts about the clinical issues, the practical issues, and also for developers who would like to continue this method, I will provide technical content as well. Okay, so as you know, we have the alpha motor neuron, and the pulses from alpha motor neuron is transmitted to muscle fibers, and it innervates the muscle fibers through motor end plates. So it is the procedure of first generation. And well, when we put needle into the muscle, and when we have this motor neuron junction, the propagation of the action potential through the muscle fibers will reach to the electrode, and the electrode picks up the voltage, and you will see something like this in your computer, which is called motor unit action potential, with the action potential that is generated by the motor unit and is caught by the electrode. Usually we have signal, and we have the analog signal. Then we also have a filter that usually is the anti-aliasing filter to reduce the bandwidth of the signal, and the signal is transferred to the discrete time signal, and it is stored in the computer. When you look at the signal in the device, it is a wideband signal, as you see here. But as you can see here, the bandwidth of the signal contains a lot of information. Suppose that you record signal by 10 kilohertz sampling rate. So the bandwidth of the signal is limited to 5 kilohertz, and this analog filter, which is called anti-aliasing filter, limits the frequency in order to have good signal for processing. So from 5 to 10 hertz, usually we have the electrode drift, the half cell drift potential, and we have to get rid of it because it doesn't have enough information for us to precisely locate the action potential of motor unit. From 10 hertz to 5 kilohertz, we have information. However, around 10 hertz to about 1 kilohertz, we have the volume conducted components of motor unit action potentials for those motor units that are far from the recording electrode. And since we have the volume conductor, we have the decrease of the amplitude and also the frequency of the contents of action potential that are related to motor units far from the electrode. So they cannot be used to precisely locate the action potential. So in decomposition, in intramuscular decomposition, we usually use a high-pass filter with the cutoff frequency of between 500 Hertz to 1 kilohertz. And then we have the spikes of nearby motor unit action potentials from 1 kilohertz to 5 kilohertz. It is important to mention that we also have thermal noise all over the place. So when we get rid of this part, we not only get rid of the action potential of far motor units, we also reduce the amount of the energy of the thermal noise. So this is what we do before doing any processing, which is called the pre-processing or signal conditioning. Well, when we filter signal with a high-pass filter, we get something like this. And if we decompose it, we will get something like this. So we need to identify each motor unit and each time that the motor unit fires. So it is the process of decomposition. So we have a composite EMG signal, which is also called the interference pattern. And through decomposition, we get the motor unit action potential trains. So this is the ultimate goal of any decomposition program. So the EMG decomposition has various outcomes from clinical and physiological perspective. So from the physiological perspective, it is related to the way we get the output. If we have the complete pattern, then we will be able to identify synchronization. We will be able to identify motor unit patterns. We can identify various physiological phenomena like motor unit driving effect or onion effect or any other physiological concepts. So we need to have a complete output in order to reach the best possible physiological results to be able to use in clinical practice. So this is the segmentation. After we filter the signal with the high-pass filter, we need to identify locations where any active segment, I would say, any activity of nearby motor unit exists. So we need to use a kind of segmentation or signal detection in order to identify them and discriminate them between the background noise. We need the background noise. I will talk about it and how it is important. But for now, we are focusing on those active segments that has the activity of one or more motor unit to analyze. When we do the decomposition, we will able to identify the motor units. We will able to identify, the occurrence of each unit during time. We will able to identify the shape of the motor unit, and then we can we can go back to the raw signal, and we can even extract morphological information from the raw signal. So that's why the decomposition is very important. So what we will get is something like this. And I will go through this procedure technically and clinically afterwards. So we would like to reach the templates. We want to know where they are located. We want to check the firing pattern of each unit and all the unit together in order to identify like the common synaptic inputs, the common driving effect, and anything anything in which the activity of units are required. We also need to identify the accuracy of the decomposition, which is very important because when you decompose a signal, especially when the signal is complex, you really need to know if the signal decomposition is valid. So it is a concept of validation, and a lot of people have been working on this, not only for intramuscular, but also for surface signal decomposition. So it is also an indispensable part of decomposition. So when you do the decomposition, you have to identify the accuracy for this. So we not only developed the EMGLab to do the decomposition in an interactive way, we also implemented a method to identify the accuracy of this. So it is a plugin for the decomposition, and it is available. And I think the paper was published in 2011 in IEEE. So we also have the MATLAB function for this. So if you would like to assess the accuracy of our decomposition, you are welcome to use this. So you can contact us and receive the routine if you don't have it. So we have the EMGLab program, and it is basically developed by Kevin. So I remember 2001 when I was working on my master's thesis on EMG decomposition, and I was in contact with Kevin. So we were talking about the procedure of automating decomposition to have a better dissemination. So we started working on this, and a lot of different versions were produced, and this is one of the mature version. We have a lot more version for multi-channel decomposition, for accuracy assessment, but what is available now is this view that in which you can see the signal, you can see the residual of the signal after decomposition, which is very important. You can see the templates, those motor action potentials, and the trains here, and you will be able to manually modify it or correct it, because it is very important to do this. So I'm going through this template matching technique from the signal. So let's focus on the problem. Suppose that we have a filtered signal. You can see from 100 millisecond to 200 millisecond, and then another epoch and another epoch. We have 3 epochs. When a clinician would like to decompose it, the clinician looks at the signal and also hears the voice of the signal. It is because when you hear to the voice, you are able to identify if the contraction is constant, if something's wrong, or if you need to put a needle in another place. So it is a very important section of the recording, as all of the clinicians do know, those who work on neuromuscular disorders, those who work on conduction velocity, people who work with needle, they look at the signal and they hear to the voice. So they tune the needle based on the feedback they have to be able to pick up the highest signal to noise ratio and in the best possible place, along with the standards that exist in clinical practice. So suppose that we recorded a good signal, okay? A good signal in terms of SNR, the location of the electrode, and now you would like to decompose it. And now suppose that you would like to do it manually. I talk about it from the manually point of view, and then we automate it through template matching. We look at the signal. We look at the signal and we are trying to identify similar shapes that could be possibly action potential of a motor unit. Okay, so at the first place, I look at this one, and I will get some information. You know, by looking at the signal, we get various techniques, and we implement it in our manual or automatic decomposition. First of all, we look and we say, okay, let's focus on those that have higher amplitude, because I might able to see those with lower amplitude, but it's tricky. And and then I have the activity of some units together, then these large ones will overlap these ones, and I'm not able to identify this. In other words, when the amplitude of the active parts are similar to the background noise, I prefer to start decomposition for those that have higher SNR. So for this, I take this one, I just slide it over the signal by my eyes, and I see, okay, I see something similar, right? So they might be a cluster, but I'm not sure yet. So I move my window and I see, wow, there is another one. So in these 3 lines, I'm able to identify 3 occurrences, and possibly it's a motor unit. Why I say possibly it's a motor unit? Because I need to have more frequent items of this through the signal. Let's say considering the minimum firing rate of 5 Hertz. Okay, so I need to have at least 5 of them in 1 second signal. So I slide this window through the signal and I try to locate them. So, and I make them as one cluster. Okay, so it's a cluster 1, which is in a red one. I continue and I see, okay, there is another one, and I can differentiate it by others because of the existence of the background noise, which is not that bad. The noise is not really high, so I'm able to locate the shape of the unit. And then I slide, I pick it as a template, and I try to match it through the signal. So it's my template, and I match it through the signal, and I see, wow, it's a high correlation, another one, and then I match it again, and I see something that might be this one, but the problem is that some parts of this is overlapped by the activity of other motor units. But so far I have 3. I continue. Well, I try to go to the other one, which is the green one. This one, and I match it. This is another one, and this is another one. So I will have 3 clusters. And this is cluster 4. Here, we move it here, and I try to locate it here. Possibly it would be here, because in a normal, as you know, when you have constant posture, constant force, isometric contraction, and for a person who is healthy and it's not at the very beginning or at the end of the contraction, they tend to be regular. So if it fires here, if it fires here, it might fires here, it might fire here, okay? But for now, since I'm not sure, I'm not trying to match it here, but I rather use another approach. So what's this approach? It is this approach. Well, those units that were already identified are subtracted from the signal, and I get the residual signal. So, for example, this one, I identified this. So it's a template. So I reduce it from the background signal, and I identified the residual signal. So for all of the units that were already identified, now we have the residual signal. So when the residual signal is uniform, I would say I cannot find the nearby motor unit action potential where I am confident about it. But for others, now they can be seen better way. For example, this one, okay? This one is here and also here. This one is here and also here. So when you subtract the identified units, you will be able to extract other units from the residual signal. And here it comes. When you do it one by one, you will get the complete decomposition. So you identify the location of each unit. You will be able to look at the background or residual signal to see if you miss something. You will be able to get the template of the units, from which, if you get back to the unfiltered signal, you can even identify the length of the muscle fiber, the orientation, and a lot of information. Okay, and also the firing time. From the firing time point of view, you can identify the regularity, and with the EMGLab that you will see later in another session, it is possible to extract other information from this. So here is the complete decomposition from, the 3 epochs of the signal. You can see the location of all of them, and here 1 and 8 overlapped, and now the residual is minimized, and also 1 and 2 overlapped, as I mentioned, and now the residual is minimized. So when you look at the residual signal, the brown one, you will be able to see if you already missed anything or not. So it seems nice, but we need to assess the accuracy of the decomposition, which is not part of this lecture, but I have to say that it is very important to make sure that your results are correct. In other words, there shouldn't be any alternative decomposition to have reduced residual signal. Otherwise, that one is a better one. Okay? So how can we automate this? So that was from the practical point of view. And now we are focusing on this automation. Okay. First of all, I have to say that the template matching is one of the most efficient algorithms for intramuscular EMG decomposition. It has been used possibly in 1974, and by now there have been a lot of algorithms in the literature. We also design density-based clustering and other types of fuzzy clustering and these kinds of stuff. But I have to say that this is the template-based clustering is one of the most efficient one if we can solve some minor problems. Let's see what we have. Well, what we do is that basically we start by checking each active segment, and if this active segment is the first one. Okay, sorry.
Oh, I think it was a mistake. You can continue.
Yeah. Okay, continue. Okay, sorry. Okay. Okay, so the first segment is considered as a sphere.
You are muted, Hamid. You are muted. Can you unmute yourself?
It's the program.
Yeah.
Did this to me.
It's
a bad program. I'm here. Okay, thank you for the feedback. It's okay now?
Yes, it is. Thank you.
Okay, perfect. So how can we automate this? Let's get back to the signal and try to imitate what a human expert will do. What I do is this one. I pick the first segment. Since it's the first segment, I consider it as a cluster. Then I check the other segment. I need to check if it is similar to one of the already identified clusters or not. So I need to identify the similarity between the second segment and the first segment. If it is acceptable, then it is merged with the previous one. Otherwise, it is a new cluster. So I continue all of these procedures during time, and I try to match the new active segment to those that have been already identified. I either merge them together and then I update the cluster, since when I update the cluster, I would follow the electrode movement, because during recording, the electrode might move. So the shape would slightly differ. So when I for example, see that I have a template here, and then I try to find another part I find here, then I find a way to update the shape based on the newer information to be adaptive, you know, to be able to compensate the electrode movement. So let's get back to the automation. Here is the automated part. I provided all of these parts as a flowchart. So in this flowchart, we have the procedures of template matching. Then we also have the procedures of cluster merging, because sometimes we identify two clusters that are basically the same units. So we really need to merge them together. But there is a problem here, and it is very important to identify this. So I prefer to write it down here rather than talking with this. Let's do it with this way. Okay. The point is that if here it comes. Okay. Suppose that I have unit 1, something like this, and I have another one, something like this. That is the core item of the advanced template matching. And I'm going to provide it to you as with a working example. The point is that this is the new one, okay? Basically, if I want to identify if it is related to this unit or this unit, I need to identify the similarity between these two and the similarity between these two, okay? So we need to identify the similarity. Similarity. That can be, for example, the Euclidean norm or correlation or any other indices that can identify the correlation. On the other hand, how can I make sure that these two are similar enough? We need a threshold, right? So I would say if similarity between the new one and unit 1 is higher than a threshold, then assign it, right? So this is the problem. I would call it a problem. Why? Because we cannot use a fixed threshold. You know, if the threshold is low, then for those units that have high amplitude, it doesn't work. So if we make it as a low threshold, then nothing can be merged with this one. Since the amplitude is high, and we have some problems that I will talk about it is impossible to merge the new ones with this one. On the other hand, if the threshold is high, we will have problem for those with low amplitude. So they will be merged together, because when you have low amplitude, a lot of templates would be similar. So basically, we need an adaptive one. Okay. If I solve this problem, if I can extract an adaptive methodology to extract this threshold, then I will be safe, and I will use an algorithm that can be used in real time. It is very important. Because template matching has a very low computational complexity, it can pass over the signal once. And if you correct the problem of the threshold, then you will have a method that can be used offline, online, and you do not really need to cope with the other algorithms that are more complicated, especially for the signal that has all the information in the shape. So unlike other signals, intramuscular signals has information, complete information from the shape of the units. So what can I do? How can I make it an adaptive threshold? Okay. So here is what we do. Let's get back to the sampling problem. And I identify the approach here. Okay. You know, the red signal is the continuous signal. But when I sample it and I record it in my computer, I will have some points, 0.1, 0.2, 0.3, 0.4, etc. And since the sampling procedure is not synchronized with the peak of the waveform, it is possible that when I pass through the signal and I see something similar, I might sample it from other locations, okay? So basically, when a human operator look at the signal in the continuous way, these two are similar. But when you have the recorded signal, just those samples, since your sampling is not synchronized with the peak of the waveform, and since your clock is not completely 100%, I would say 100% accurate, you also have some fluctuation. Then when you get the error between these two, although you know they are similar, you get some error value, okay? So one way of the problem is for the sampling. I would say time quantization, okay? We have time quantization error. So this is one of the errors that can be seen in the automatic template matching if you want to do it automatically. So you have to somehow compensate this one. So this is one source of the error. The second source of the error is background information. I would call it background noise. Okay, so when you have signal with different background noise, you would say that it might affect what you are doing. So example, if this is the background noise, it would affect because it is summed up to this part too. So it's like a noise everywhere. So this is what will affect your alignment and your template matching procedure. So the higher the noise, the higher variability you will expect. Okay, so when I get back to this one, I would say that I have background noise, and the lower the SNR, the higher variability you will expect. Okay, so what we can do for each template, we identify personalized threshold. Okay, so the threshold would be personalized, and it is the sum of the variability that we expect from part A and from part B. So from a statistical point of view, we extract background information. We extract background information from those those segments that do not have any activity. Okay? And from this, we extract the variance of the noise. And from the variance of the noise, we are able to extract the amount of variability based on the statistical methods that you would expect. On the other hand, for this, what we will do for each template, we identify 0.4 sampling interval where we can expect the variability between shape. For example, it will cover this one, it will cover this one, it will cover this one. So what kinds of different trajectory and shapes you expect to be visually acceptable in order to be assigned to a template. So for this one, which is based on the high resolution sampling, we extract another variability that is related to this specific unit. So we have variability 1 and 2, and they can be summed together because the background noise can be seen as an additive noise compared to this one. So we have a threshold that is the combination of variability 1 and variability 2. So for each unit, when we have a new segment, we compare, if we want to compare with this, we calculate the similarity and then we measure with this threshold. So in this case, we will get rid of the problem that I was talking about. So let's get back to this. Here. The similarity must be above a threshold, okay? You cannot simply say that, okay, we have 5 already identified units and we have a new active segments. Unit number 3 is the closest one, okay? It is the closest one, but is the similarity over the threshold? If it is over the threshold, we assign it. Otherwise, we don't assign it. There have been a lot of work in the literature talking about this. That's why they propose fuzzy based clustering or other kinds of clustering to consider all of these things that are not completely fixed. So here we identify a threshold which is not fixed, and I talked about it, and is adaptive because it is measured from each signal differently. So a signal with different SNR will have different threshold. And we even update the threshold during time. So for example, suppose that this is the background noise, and then suddenly we have a higher noise, and then we have a lower noise, okay? So let's say for each 1 second, the threshold one is identified, and it is updated. It is updated to get the second threshold, a third threshold, etc. So, and for example, if a unit, then as I mentioned, we update the templates during time. So basically, the second threshold will be updated during time. So both of these thresholds will be updated during time to compensate the background noise that is changing and the electrode movement, etc. Okay. If we also include some kinds of firing time information, then we call it firing time inconsistency, we will able to incorporate the firing time information as well as an active way of clustering. But now we just focused on the template-based method. When we have signal from different channels, it would be easier because it's like we have signal in multi-dimensional space. So when one template is very low in one channel, it would be higher in another channel. So we find those channels for those segments that have higher amplitude, and we try to use them for template matching in different channel, and then we combine them together in order to increase the accuracy of the composition. That's what that was my presentation, and I hope that it was suitable for you, and I'm ready if you have any question or to answer. Thank you very much.